Okay, good morning everyone. Thanks to those of you that have made the effort to come today on a Thursday. I appreciate that. I'm going to talk today about the thing I'm probably most interested in scientifically, and I could talk about this for hours, but I'll try to stay within the 45-minute allocation for the lecture. But of course what we've been talking about, and not only in lectures but simultaneously trying to pursue in our lab course at the moment with our drug discovery projects, is of course we're very concerned about this phenomenon of oxidative stress, where we get this overproduction of reactive oxygen species, usually as a result of some sort of endogenous process. And this can result in direct cell damage, which is hypothesised to play a role in disease. And you can probably find papers somewhere published by someone implicating reactive oxygen with about any disease you can think of. So that's how widely influential this hypothesis has been. But when it comes to pharmacology, obviously we want to block pathological processes. So one of the most obvious approaches is to use a direct acting antioxidant to try and counteract reactive oxygen species within the cell and block their contribution to disease. But the problem with that, as we highlighted in our lecture on Tuesday, is that we didn't appreciate that free radicals are actually quite important, or playing quite important beneficial roles in physiological systems. And so one of the things that happens when you take mega doses of vitamins is you might produce some unexpected side effects, things like infertility in blokes and vulnerability to infections, because reactive oxygen species are very important in mediating the effective roles of the immune system in eradicating pathogens. Okay, so that hypothesis has sort of struggled, and there are a couple of drugs that have really, only edaravone's really made it onto the market as an antioxidant, and I actually believe it's not an antioxidant. I actually believe it's an acrolein trapper, and you'll probably join me in believing that, hopefully after next week's labs, because we're going to use it as a carbonyl trapper. Okay, so what I'm particularly interested in is, okay, why don't we try directing our drug discovery efforts against some of these nasty aldehydes formed during lipid peroxidation? Because remember that unsaturated bonds, particularly in polyunsaturated fatty acids, are very vulnerable to attack by reactive oxygen species, and this triggers the production of some really nasty reactive aldehydes, and the one I'm particularly interested in is this little 3-carbon compound acrolein, highly noxious and toxic. And so on this strategy then, since these are so chemically reactive and prone to form protein adducts, perhaps we can identify drugs that trap these carbonyl products or these aldehydes without and suppress their role in cell damage, and hopefully not have too much effects on physiological roles of antioxidants. Now this is easier to draw on a PowerPoint slide than to achieve in an actual molecule, because it turns out the chemical properties of a molecule that make it a good antioxidant often make it an aldehyde trapper, so, and vice versa. So separating carbonyl trapping activity from an effect on trapping superoxide radicals, for example, is not as easy to achieve as we might imagine. But nevertheless, that's the goal of what we're trying to do. Okay, so what we've been emphasising through these lectures is that the indiscriminate nature of the chemical damage caused by reactive oxygen species produces a real soup of nasty species in cellular environments, and this is particularly manifest in the production of reactive electrophilic aldehydes during free radical damage to cell membranes. And we call this phenomenon carbonyl stress. So it basically accompanies oxidative stress, and these are non-radical species that are very electrophilic, and that means they want to react with a nucleophilic amine, say on a lysine or in a histidine ring, or a sulfhydryl group on cysteine, highly prone to modification by these sorts of nasty species. Okay, so we can define carbonyl stress as a heightened cellular exposure to diverse reactive carbonyl containing electrophiles and formed during fragmentation of particularly unsaturated lipids. Okay, so we're trying to trap these nasty species in cells with drugs to prevent them from attacking proteins and other critical cellular components. Now, as far as I can tell from the literature, the first person to really think of this idea was a chap called Shapiro. That's where I found the idea and encountered it 25 years ago, which shows you how old I'm getting, in the American Journal of Therapeutics. And he made the suggestion that opportunities exist for therapeutic invention by use of certain water-soluble small molecular weight drugs that contain primary amine groups. Such pharmaceutical agents administered orally can form shift-based derivatives with toxic carbonyl substances and thus protect cellular components. So that was a good idea. And the idea he had is using an amine-based drug to trap the carbonyl group, because this is a well-documented type of reactivity of a carbonyl compound. It forms what we call a shift base. And rather than this happening on a protein, by administering a small nucleophilic drug, you can trap it with trap the carbonyl with a drug. Okay, the problem with this though, and we're going to come to this, you'll see this in my pre-lab that I'm working on at the moment, hopefully I'll put it up today, is with acrolein, this is a bit tricky because the oxygen isn't the most reactive part of the molecule, it's actually the double bond. So that means using an amine-based trapper might not be the best approach. Now we're going to use one in our lab class, but one of the compounds in our library is an amine-based molecule. We'll come to that in a minute just to highlight the possible chemistry of that molecule, but bear in mind that this may not be optimum using an amine-based drug. Now that doesn't stop some people rushing into research and drug discovery and development with an amine-based drug. And if you want to have a look online, this company in the United States, Aldera, is focused entirely on this sort of carbonyl trapping pharmacology. And their lead compound, reproxalat, you can see it contains an amine here which they're relying on to act as a aldehyde trapper. And they've reported some nice data recently. They're doing some phase 2 clinical trials. So remember, a phase 2 clinical trial is with people with a disease of interest. So you're trying to see whether your drug works or not on a relatively small scale before you scale up, go to a large phase 3 trial. But they're particularly interested in ocular diseases. And this is an interesting idea because by going after diseases of the eye, you can administer the drug to the eye and you don't have to worry about oral bioavailability issues as you do with an orally administered pharmaceutical. And so one of the conditions that they're getting positive results with is dry eye disease, this unpleasant sort of condition that affects people. And you can see with this particular compound, reproxalat, they see this reduction in a number of the symptoms, unpleasant symptoms and signs of dry eye disease. So this is quite encouraging. It's really the first positive clinical data for a carbonyl trapper. Note that they use a different terminology to me. They call it RASPI, a reactive aldehyde species inhibitor, but it's basically the same idea. Okay, so this is interesting, conclusion, proof of that hypothesis of Shapiro 25 years ago that a drug might be able to do this. Now, another area where people are getting some interesting results is to use drugs directed against another nasty type of another family of nasty carbonyl compounds, and this is one we haven't actually worked on, but these are, and I think I highlighted this in my lecture the other day, that one of the products of lipid peroxidation is this nasty 9-carbon substance called ONE, oxononenal. And the nasty thing about this, it's a dicarbonyl, so it's got 2 carbonyl groups. And they're actually called gamma dicarbonyls, because as I explained in my tute coming up this afternoon for you, is chemists name things with Greek letters according to where the substituents are. So this is a gamma dicarbonyl because the 2 carbonyl groups are located by 3 positions apart. So that's an alpha, that's a beta, and that's a gamma. So the 2 dicarbonyl groups, or carbonyl groups, are located in gamma orientations. Now these are really, really nasty and really, really reactive. And they are especially reactive with amine groups on protein, so they behave as hard electrophiles. Remember how I talked about that in my toxicology lectures last semester? Hard electrophiles react with nitrogen and oxygen, soft electrophiles react with sulfur. Okay, so these things react extremely rapidly with protein amine groups, and they form these unusual pyrrole adducts. So this new 5-membered ring, don't worry too much about the chemistry, but it's just a 5-membered ring forming as a result of that big lipid structure cyclising on the nitrogen. So this other form of dicarbonyls, in addition to ONE, is isolevulglandin. These are extremely nasty and reactive. They form from arachidonic acid, but there's no fragmentation of the arachidonic acid molecule, so the whole 22 carbons get stuck on the protein, and it forms this 5-membered ring, and the remaining carbons are hanging off these side chains. So they're quite bulky adducts. And a group that's been working on these in a lot of detail at Vanderbilt University, they've found these adducts in a whole variety of diseases, including some significant cardiovascular conditions. So sort of watch this space, because I think this area is really growing in importance. And I've also tested a number of compounds as inhibitors of, or trappers of, these nasty dicarbonyl electrophiles. And they're pretty simple molecules, really simple molecules, just really, most of them are ethylamino aromatic compounds, pyridoxamine here, same sort of feature here, an ethyl amino substituent. N-acetyl lysine works pretty well, 5-methyl-2-hoba. 2-hoba. This one they're really getting some interesting results in, and they use this in clinical trials. You can buy this from health food shops. It's available as a nutraceutical, so it's not subjected to the same proof as a FDA type approval. And I'll talk about that in a minute. But the one we're looking at on, if you notice in our chemical library, we've actually included one of these compounds as our putative carbonyl trapper. So HOBA, 4-hydroxybenzylamine, is our compound, UWA 0012. And no one seems to have tested this compound against acrolein-mediated protein damage, so it'll be very interesting to see how it goes in our labs next week. Okay, so what do these things do? Well, here's 2-hoba. We're using 4-hoba, but 2-hoba forms this that same 5-membered pyrrole type substituent group from trapping the whole oxidised isolevulglandin, the whole oxidised arachidonic acid, and inactivating it. Okay, so these groups have published some nice reviews in recent times from pharmacology at Vanderbilt in annual review of pharmacology and toxicology, and also pharmacology and therapeutics the last couple of years. And these are high-ranking pharmacology journals. It's sort of nice that these reviews are being published, because it means that this idea of trapping these reactive oxidised lipids and their fragments is becoming more mainstream within pharmacology. So that's good news for all of us. Now, but I'm particularly interested in these short chain toxic products of lipid peroxidation. And of course, acrolein is the one that's particularly interesting from my point of view, but there are other chemically related molecules as well that behave with similar chemistry. Okay, so these are interesting, and I talk about this in my pre-lab e-shoot, explain this a bit more, but it's the closeness, this proximity of this double bond to this carbonyl or aldehyde group that makes this a really nasty combination of chemistries together, and it makes this double bond really, really reactive. So this is why acrolein is so toxic and why the Germans and Pommies bombed each other with it in World War I, because it is so nasty and it's very reactive towards the epithelium of the respiratory tract if you inhale it. And so it's a big player in smoke inhalation injury, for example. People who get trapped in burning buildings these days, most of the people that die from burn injuries, it's not from the dermal burns. We've gotten very good at treating that. The burns to damage to the skin, you can usually survive, but what often kills people is smoke inhalation injury. So inhaling smoke is really bad for victims of burns, forest fires and domestic fires, that sort of thing. So kills off the particularly the integrity of the lung, and it becomes waterlogged basically. Fluid floods into the lung from the vasculature because of the damage caused by acrolein. Okay, so this is becoming more of a problem because even just sitting in this lecture theatre now, we're sitting on these synthetic polymers in upholstery, and when in our carpets made out of synthetic polymers, and when synthetic polymers burn, they release a lot of acrolein. So this is why this is becoming more of a problem in the modern world. There's also lots of acrolein in tobacco smoke, so it's a big player in chronic obstructive pulmonary disease associated with tobacco smoking. Okay, so where does acrolein come from? Well, the chemistry is complex, but it's basically thought to arise via cleavage of some of these oxidised lipids, rearrangement of them. Cleavage of these hydroperoxide species liberates acrolein during lipid peroxidation. Now, acrolein is also important in medicine, and one of the classic anti-cancer drugs you might have heard about in previous lectures, probably from David Joyce, hopefully, is cyclophosphamide, a broad purpose anti-cancer drug. One of the problems with using cyclophosphamide, you have to watch the urine output of an individual because they can experience nasty bladder injury, and it's actually caused by formation of acrolein within the bladder via a cytochrome P450 product. So to counteract that, a drug has been used in medicine for a long time known as Mesner, and this is a good acrolein trapper. Now, I talk about this in my pre-lab at the moment in more detail, but the reason acrolein is so toxic is, as I mentioned, that double bond becomes very reactive because the nearby oxygen pulls electrons away from the double bond, makes it very reactive with a nucleophilic nitrogen or sulfur. And so what happens, acrolein reacts with proteins very readily, and the dominant adducts are what we call propanol species, and these retain a carbonyl group. So this is on lysine. The one that's most quantitatively significant occurs on cysteine, because this double bond in acrolein reacts as a soft electrophile, so it reacts with a soft nucleophile in the form of the cysteine sulfur. Very, very fast reaction, so we get lots of carbonylation adducts. Also occurs on histidine and even to some degree on arginine, but the point is that there's a lot of these adducts retain carbonyl groups. That's a dominant pathway of protein damage. So that's what we're making use of in our lab class next week. We're going to use 2,4-dinitrophenylhydrazine to trap these carbonyl adducts formed by acrolein in the protein. So hopefully any of the compounds in our library that are effective will block the formation of these adducts by acrolein. But it also means we can pull out proteins using these probes. Remember I talked about this in my lecture on Alzheimer's last week. Biotin hydrazide is particularly popular because you can pull all the biotin labelled carbonyl adducts out of a protein mixture with a streptavidin system, chromatography, and then fire the proteins into a mass spectrometer. Now there's not a lot of labs doing this sort of stuff. It's sort of a shame because it is quite challenging to pick up the low levels of adducts on proteins. Is actually pretty demanding mass spectrometry. But one of the pioneers in this field, Dan Liebler, did some very nice work in this area where he used a slightly different type of probe, an aldehyde reactive probe, where he used a sort of methoxyamine type conjugate here. I can't remember what that's called, but it traps aldehyde containing proteins, and then you can pull them out because it's got this, I think it's a biotin tag there. And when they did that, they used this method just to look at completely healthy mitochondria pulled out of the heart tissue of rats. And I thought the results were amazing because they looked at where the adducts forming, and most of them are forming on cysteine. 85% of the carbonyl adductive proteins come from cysteine. And then the mass spectrometer allows you to determine what is the aldehyde stuck on the cysteine group, and look at that acrolein lights up as a major player. So some of these other carbonyl compounds are much less significant, but 5 out of 6 adducts or so were acrolein derived. So this is one of the reasons. So this is published quite a while ago. Unfortunately, Liebler retired not long after publishing this paper. But it's a real shame because there's not many labs around doing this sort of stuff, but it would have been really nice to use this technology to get insight into what's going on in diseased tissues. Plenty of papers just measuring a single aldehyde in a particular disease, but this is the first time anyone's tried to look at a whole bunch of them and work out which ones are important. Okay, so this raises a question that's a real challenge in carbonyl trapping chemistry, is the fact that we're getting such a soup of electrophilic species being formed. There's going to be a lot of chemical diversity in terms of their reaction with proteins. So, like I said, a lot of the focus has been on these hard electrophilic aldehydes or carbonyl compounds like isolevuglandins. And remember I said they react really extensively with lysine and histidine perhaps. But acrolein a bit more challenging because while it does react with lysine and histidine, its preferred target is that soft thiol group because it's got that soft electrophilic centre in that double bond. It wants to react with a soft nucleophile. So that means that a trapper that traps a hard electrophile like isolevuglandins may not actually be that good against acrolein. So this is one of the things that our research is pursuing. And that actually holds true when you look at what are some of the compounds available that are used to trap acrolein. And this is one of the challenges in this area because things like mesna that I mentioned is used to treat patients at risk of cyclophosphamide urotoxicity, bladder toxicity. It's a sulfur compound. So too is N-acetylcysteine. It's used to treat paracetamol poisoning. It traps electrophilic soft electrophiles derived from paracetamol napkin. It's not the same as acrolein, of course, but it's still a soft electrophile. And again, it's a sulfhydryl group there doing the trapping. Dimercaprol. You can find literature in the showing that that protects against acrolein toxicity. It's a thiol compound. It's used to treat heavy metal intoxication. D-penicillamine, used to treat rheumatoid arthritis occasionally. It's a bit toxic. Also used to treat heavy metal intoxication. Again, another sulfhydryl compound. And look how all these molecules are delivered to patients, parenteral. You can't give them orally. Thiol compounds are very vulnerable to metabolism by P450. They have very short half-life, low oral availability. So that's why you have to give them via an intravenous route. So that's not very desirable for a carbonyl trapper. As far as we're concerned, we want to identify an orally active carbonyl trapper. And so we spent a long time working on hydralazine. And this is a different chemistry because hydralazine contains a hydrazine out here, and that terminal nitrogen there is very nucleophilic. But it's got some issues because it causes a drop in blood pressure, and that's the primary clinical usage of hydralazine, to cause vasodilatation and treat high blood pressure. Now, one of the reasons we're interested in going after acrolein is that the evidence seems to be accumulating that this is a nasty little species that contributes to some serious diseases. And I mentioned alcoholic liver disease a couple of lectures ago, and interestingly, a group in Kentucky published some work back in 2016 showing that acrolein, they reckon, is a major player in alcoholic liver disease. And remember I mentioned that work in my lecture a couple of lectures ago. They found that the urinary marker, the HPMA, a metabolite of acrolein, a mecapturate formed from acrolein metabolism, excretion in urine, is a good predictor of severe forms of alcoholic liver disease. And this raises a question: can a drug like that traps acrolein be useful in treating alcoholic liver disease? And so this is, we did a lot of work on hydralazine, probably wasted too much time and money on it really, but we found it's an excellent acrolein trapper, and we identified this product here, EAPH. But then we also found a problem with this drug is that it reacts with acrolein adducts on proteins. We call this adduct trapping, and this is sort of bad news. It might actually be beneficial because it might so-called cap the acrolein adduct and stop it from cross-linking with other proteins. But this problem arises because hydralazine traps the wrong end of the acrolein molecule, and we'll come back to that in a minute. But anyway, this group in Kentucky, they read our papers on hydralazine. They thought, oh, I wonder if this works in an alcoholic liver disease model. And so they gave it to mice that were subjected to a chronic binge alcohol feeding model, where they get, it's sort of hard to get mice to drink enough alcohol to show signs of alcoholic liver disease. Only humans are silly enough to drink massive enough quantities of alcohol over a long enough period to get alcoholic liver disease. But if you put it in a liquid diet with corn oil, along with ethanol, so that they're getting 35% of their calories from this ethanol source, it's a pretty high level. And if you give them that for 10 days and then you give them a single oral gavage of ethanol, you can reproduce a lot of signs of alcoholic liver disease in an animal model. So short-term model, but it reproduces the sort of liver injury you see in humans that's associated with binge drinking. And when you do this, they found that there is lots of acrolein adduction occurring in these ethanol-treated animals compared to controls, often central lobular around the central vein. But then if you put the animals on hydralazine, this was a daily dose of hydralazine, you could block the acrolein adduction occurring. You also protected against some of the overt liver damage in the form of vacuolation, some of these other pathological changes detected with H&E staining. You also reduce fatty liver, classic outcome of alcoholic liver disease with hydralazine treatment, looking much more like controls. And then you also reduced apoptosis occurring, detected with a tunnel assay. And then you also reduce the serum enzymes, liver enzymes floating around in serum, although the results aren't too stunning. But certainly suggests that hydralazine as an acrolein trapper does hold promise for, or at least it does show that an acrolein trapping drug does have promising potential in this sort of model. Now, another disease where I think acrolein is emerging as a really significant player is in spinal cord injury, and this group at Purdue University in Indiana, led by Rishi, has done some very nice work over the last 20 years looking at acrolein as a diffusible mediator of spinal cord injury. And so what people often don't appreciate is that if you have a traumatic injury to your spinal cord, let's say a car accident or a sporting injury, there's a primary lesion to the bundle of nerves that's occurring, but then over the next couple of weeks you get this pathophysiology occurring as the injury spreads up and down away from the spinal cord. And this has raised the interest of people like myself, because there's a lot of lipid peroxidation occurring, and maybe it's these diffusible aldehydes making their way in both directions from the lesion site that's actually killing off the spinal cord neurons. Okay, now there's lots of stuff going on in a damaged spinal cord. It's not just all about aldehydes, but certainly the work of Rishi shows that acrylaine's a significant player, and one of the models he's used is to take mice, and it sounds like a horrible model, but you can put them under an anaesthetic and drop a weight on the spinal cord and inflict a contusion injury, similar to traumatic injury, and then they anaesthetise the animal and take the explant out and put it in culture media. And if you do that in a control animal, the spinal cord lives happily for a few days. But if you do it to a contusion injury model animal, spinal injured mouse, you get this lesion that expands over the subsequent days. And just like you see in a human model, where you see that secondary wave of spinal cord injury occurring. And interestingly, these guys have shown that in addition to the death of neurons, you actually see an increase in acrolein adducts forming as a result of this secondary process of neurodegeneration. And the cool thing they've done, I thought was pretty cool, they took a spinal cord explant from an injured mouse and co-incubated it in the same well with one from a healthy control mouse, and they were able to show that acrolein was diffusing out of the damaged spinal cord, and you could see acrolein adducts forming on the healthy control spinal cord, and also neurodegeneration was occurring as well, death of the neurons. So this is just a nice little model for looking at the role of diffusible products in spinal cord injury. And they also showed that if you put good old hydralazine in there, you could preserve the control spinal cord explant. So that's a sort of nice hypothesis to show that some of these products being released from traumatically damaged tissue can diffuse out of the tissue and damage healthy tissue next to it. Now, the problem though with using hydralazine, this is what I want to wrap up with, is it's not the ideal acrolein trapper for a number of reasons, and that's why we sort of stopped work on this molecule after publishing a dozen or more papers on it. Now the reason it's interesting is because it contains this very nucleophilic nitrogen on this hydrazine group, and it actually acts as a carbonyl trapper normally. If you take hydralazine as a healthy person, a good proportion of the oral dose would come out in your urine as a pyruvic acid metabolite. So pyruvate has a carbonyl group and it gets trapped by hydralazine as it goes through the liver, and a significant proportion of an orally administered dose, I think it's about a third or 35%, 40%, if I remember rightly, derived from this trapping adduct. So that sort of proves that in theory that a drug can trap reactive carbonyls in the body, which is sort of nice proof. But the other problem with this drug is that it has this high blood pressure lowering property. And so you don't necessarily want that if you're going to treat a patient, say, with traumatic spinal cord injury. You don't want, I mean, they do have problems with high blood pressure sometimes, but nevertheless you don't really want to give a drug that's going to cause a drop in blood pressure to an acutely injured patient. But even bigger than that problem is it traps the wrong end of the acrolein molecule. And for that reason, so if we come to acrolein, remember I said It's a bifunctional electrophile. There are two electrophilic groups or sites in the acrolein molecule, but they're actually chemically different, dissimilar. So the one that's most reactive is this double bond, and I'll talk about this in my e-tute. You'll learn more about it. But this is very reactive. This beta carbon here wants to react with a sulfur nucleophile, such as a cysteine sulfhydryl group. So that behaves as a soft electrophile, but the other electrophilic centre is an oxygen, this carbonyl, which reacts as a hard electrophile. So it wants to react with nitrogen groups, such as lysine and hydrazines. But also hydralazine is essentially a hard nucleophile. So when it traps acrolein, it does so by reacting with this oxygen, but it doesn't react with that double bond. So this is an inherent limitation to using hydralazine as an acrolein trapper. It can't compete with this really fast reaction of a sulfur group with the double bond. And we actually see that experimentally when we've done studies in cultured cells. When an acrolein reacts with a carbonyl group, or to form a carbonyl adduct, it primarily reacts via the cysteine sulfur group, and that forms a carbonyl adduct. And hydralazine just simply can't compete for that really rapid reaction. And we see that in this cellular model where we treat cells with acrolein, we get a loss of ATP indicating cell death, and hydralazine protects it against cell death very nicely, but it doesn't protect against damage to the intermediate filaments. So intermediate filaments are just easily extracted from cells, and they're preferred targets for acrolein. And you can see all these carbonyl adducts forming on these keratins and vimentin, very vulnerable to adduction. And hydralazine really only blocks at very high non-physiological, non-pharmacological concentrations. 100 micromolar, you get a bit of suppression, and 300. But even at that top concentration, you're not protecting vimentin. So this was a bit disappointing because it means that whatever hydralazine is doing to stop the cells dying, it's not stopping damage to cellular proteins. So it's probably doing something else, and it's trapping adducts. So here we have an antibody we made against a drug stuck on the aldehyde, stuck on a protein, and you can see it's the drug is forming a lot of these trapped adducts. So we think it's probably stopping some forms of cross-linking, but it doesn't stop cross-linking of vimentin, which was a bit of a disappointment to us. Vimentin down here with our Western blotting forms a double a dimer after it gets cross-linked by acrolein, but hydralazine doesn't really stop that. But there's some interesting chemistry going on here because low concentrations of hydralazine increase this high mass species up here, which we never identified. But one of our theories is that the hydralazine probably attracts chaperone proteins to the damaged protein, and this might be what we're seeing up here, small chaperones. But that's beyond we sort of lost interest in it when we found that this it couldn't stop cross-linking. Okay, so that led us to search for other carbonyl trappers, and so we want to stay away from a sulfur-based carbonyl trapper. We don't see much point in using a nitrogen-based trapper like that one that's been followed up in the United States by Aldera during the clinical trial on dry eye disease. They're using reproxalap. It's an amine-based trapper. It's just not going to be very good at trapping acrolein. We've actually shown that in our own experiments. It doesn't react well with acrolein at all. Hydralazine's no good too because it's got the same limitation. You're relying on an amine. It just can't compete with that really fast reaction with a sulfur. And so this is why we're now working on darivone-based molecules. And darivone is interesting. It forms a carbon-based nucleophile, and I talk about that a little bit in the yeet sheet. We're using this as a positive control for next week's experiments. We'll be comparing everything against the Daravoine. And it's interesting that when you look at the history of a Daravoine, it's used for the treatment of stroke. It was approved in Japan in 2001 for treating stroke. And Japanese researchers, and they've been working in this area for a long time, they believe acrolein is a really big driver of stroke severity because, remember I said after a stroke you get that reperfusion injury, and they reckon there's a ton of acrolein being made there, and so they can measure acrolein adducts occurring in animal models of stroke. But interestingly, a Daravoine is mainly used to treat stroke, but they don't think it works as an acrolein trapper. They think it's a free radical trapper, but I think that's wrong. Okay, that's all we're talking about in this lecture, but hopefully next week you'll see how good an acrolein trapper is in your experimental model, and you'll see why we're particularly interested in trying to do more research on a Daravoine. So this is work we're doing with Matt Piggott in chemistry. They've got a library of about 70 Daravoine analogues. Many of them they've made themselves, and together with that group in Purdue, we're trying to get funds to support our research, particularly in the spinal cord injury context. But they've also sent one of our molecules off to that group in Kentucky where they're really big on acrolein as a mediator of alcoholic liver disease. So they're doing those animal trials right now. So we'll find out whether this Daravoine nucleus is worth pursuing. So, trying to do it all on a smell of an oily rag and without much money, but it's unfortunately when you're working on an area that people don't think has got much, don't know about, that's what you've got to do. Okay, thanks very much for your attention. I hope you've enjoyed these lectures on oxidative stress, and you'll see their relevance as we go through the drug discovery project. Thanks. Vitamin C and vitamin E, that's been shown, yeah.
Yeah, but it does
in
show,
high doses, yeah.
it does show it's, oh, so is it just like in high doses? So if you just get a bit, it does. I was just wondering, what's the difference about getting as a food or getting, is it just...
That's a very good question, yeah. I think when you get it as a food, you're getting other tocopherols as well, the vegan gammas and all that. So maybe they do have additional beneficial effects we don't know about, although alpha tocopherol is the best antioxidant. That is a good question. There's probably other things in the food that are also, having beneficial effects. And I guess when you're taking just a purified single isomer in high dose, you just run the risk of adverse effects in a way that you wouldn't. You'd have to eat an awful lot of food to get the same dose, and I think that's probably the problem. So it's blocking those beneficial roles of free radicals when you take purified forms of the tocopherol.
So maybe there's like combined effects with the other.
But if you take, I think my understanding of the literature is it's taking the big doses every day for an extended period that's the problem. So if you're just taking dietary sources, that's not going to be the same dose. You get,
oh,
yeah,
okay.
yeah.
Thank
Okay.
you.
No worries. Yeah.